Morning and welcome, to the Tina's Wish second virtual researcher roundtable. We're really excited to have you all with us today. And as I was just saying to our speakers earlier, I feel like this is one of the silver linings to the pandemic. You know, how often would we have been able to take a small group out to the University of Kansas to chat in person and to see the incredible work that Doctors Godwin and Soper are doing. So I think this has allowed us to have a really unique opportunity that we might not have had in the past. So I want to thank you for joining us. And very quickly, just as a welcome and point of introduction, my name is Beverly Wolford, and I'm the Executive Director at Tina's Wish. And as most of you know, Tina's Wish was founded in memory of Tina Brosman, who was a judge on the Southern District Bankruptcy Court. And she would oftentimes say she wasn't angry for having ovarian cancer. She was angry she did not have a fighting chance. And really, that's why Tina's Wish exists, because we are here to give all women and their families a fighting chance against this disease, which in the majority of cases is diagnosed in really late stage. So our express mission is to fund scientific research for the early detection of ovarian cancer. Just two pieces of information I also wanted to share. Just wanted to let you know we are recording today's session for those who may not have been able to join us live. And then secondly, I also just wanted to put a shout out to one of our programs that we've been really excited and working about, and that's the Empower Her series. The Empower Her series is designed to educate and empower women on a monthly basis in regards to their gynecologic health. So I will put in the chat function a link to episode number 3 that will be airing on May 6th. And that topic will be the role genetics plays in gynecologic cancers and are you at risk? So I do hope you'll join us then for a really informative session. Again, only 30 minutes and you'll really receive a lot of great information. We sent out yesterday, as just a reminder, email the bios for Doctors Godwin and Soper, but I'm also going to put them into the chat function. When you review their bios, you'd realize it would take us the full half hour to share all of their incredible qualifications, but I really urge you to read and get to know our researchers in a little bit greater depth. Without any further ado, I'm going to turn today's session over to Laureen Ryan from Alvarez and Marsal, and who's also an incredible part of our leadership council at Tina's Wish. Thank you all so much for joining us.
Thank you, Beverly. And again, I'm Laureen Ryan, and I'm a managing director at Alvarez and Marsal. And as Beverly said, I serve on the Tina's Wish Leadership Council. Thank you for joining us this morning, hopefully with your coffee or favorite drink. I first became actively involved in Tina's Wish back in 2012 and helped to actually form the Leadership Council. And one of the agendas of that council is to help bring some of these smaller events like this to help educate everyone on the research that's developing and the incredible work of the organization has evolved over time. We all have friends and family that have survived or died of ovarian cancer or other types of cancer. I have a very close friend who's, I'm happy to say, an ovarian cancer survivor. As well as, have personal experience, unfortunately, with my mother who died of cancer and my mother-in-law as well. So this is a pretty personal agenda for me, but this organization has been incredible since I've joined. I can't believe it's 2012. That's a long time. So, but what really is exciting, and that's why it's wonderful to have our researchers today, is we are really on the cutting edge and on the cusp of breakthroughs to help us achieve the goal of Tina's Wish overall. So I'm very honored this morning to be your moderator of these incredible researchers. Today, we're going to hear about the life-changing work of the Tina's Wish funded researchers, Dr. Andrew Godwin from the University of Kansas Medical Center and Dr. Stephen Soper from the University of Kansas. Dr. Godwin and Soper, along with their colleague, Dr. Claudio Soto, who is at the University of Texas Health Center, Science Center in Houston, who unfortunately couldn't be with us today, are collaborating. And I think it's really important because that's a really theme in Tina's Wish is there's a lot of collaboration between researchers, and they are collaborating to develop non-invasive and highly specific blood tests or liquid biopsies for the pre-symptomatic screen and early detection of ovarian cancer, which we'll learn more about today in just a few moments. So I want to remind the audience to please feel free to type your questions into the chat box during the panel. By default, the chat box is visible to everyone. So if you prefer to just direct your question to me, so your name remains anonymous, then choose Laureen Ryan in the chat drop down before typing in your questions. During the last 5 to 10 minutes, we'll address the questions from that chat box. For those who prefer to ask their questions verbally, you can use the raise your hand function feature in the panel, and I'll call on you to ask your questions to the doctors. So we'll wrap up this session before 9:15 as promised. You can get on with your day. So welcome Dr. Godwin and Dr. Soper. Thank you so much for being with us. I'm excited to get to ask a bunch of questions to each of you. I know everyone who joined is very interested in digging into your project and its promising path. So if I could start with you, Dr. Godwin, can you please remind us why an early detection screen for ovarian cancer is so important?
I'd be happy to address that question. And I'm going to, first, I'd like to thank again the people who are joining the call today, and especially Tina's Wish, who have given us an opportunity really to do some cutting edge science and be able to do a collaborative project across 3 campuses. And because I'm a pretty visual person, I'm going to share some slides, which hopefully will help us kind of understand some of the work that we're doing and kind of answer some of the questions.
Can I just interrupt you for a second? I'm sorry, I forgot to mention. For those who want to be able to see all these slides, you might want to put your visual in speaker view. Thank you. I'm sorry.
So really, when we talk about early detection, and it really does save lives, but the issue really has to be is that the fact that very few people are diagnosed early relative to those that have distant or metastasis. So that's when the tumor actually spreads away from the original site, which in many cases is the ovary or fallopian tube. And so what basically you see here is that there is a big decline in the 5-year survival rate based on how late is detected in the course of the disease. And as I indicated, only 15% of patients tend to be diagnosed early. And the problem ends up being symptoms alone are really a poor indicator of early disease. So a lot of the work really has to be identifying ways to screen, do routine screening to find people that have increased risk for developing ovarian cancer. And the other thing I always want to point out, because we kind of lump together ovarian cancer into one category, but it really is unique. And each tumor or each woman is really unique in that sense. But there are really 3 categories of ovarian cancer: epithelial, germ cell, and sex cord stromal. And why I point that out is the latter 2 are very much curative by surgery alone. However, these epithelial forms, which we mainly study and really are looking for to develop early diagnostic tests, are really the ones that lead to complications and eventually lead to death. And so I just want to keep that in mind as we go forward. So what I'm going to talk about really are these categories of epithelial ovarian cancers with a focus on the high-grade serous, which is the most common and also the most deadly. And that's why I also say here is that when we talk about the disease, it arises from different parts of the anatomy. So if you look at looking at the ovaries and the fallopian tubes here and the uterus, is that there are different cells within the different areas of these organs that lead to different forms of the cancer. So again, just remember that ovarian cancer, even though we use it broadly, is multiple diseases. And then each patient's disease is unique in itself. What I'm going to focus mainly on today are those now that are referred to as high-grade serous ovarian cancers. And we've learned in the past, and also this is work that's being done by another researcher being funded by Tina's Wish, and that's Ronnie Drafkin, is the fact that we now understand that some of these tumors, and maybe most of these high-grade serous tumors, actually develop in the fallopian tube. and over a period of time, develop to a point where they can spread and then shed. So that's shown here. Here's the fallopian tube. The tumors form in this area called the fimbria. And what happens is when those tumors are formed and they get big enough, they can actually start to disseminate. And then those cells start to stick on the ovary, start to spread into the peritoneal cavity. And that's why you get widespread disease. So these tumors remain hidden for a long period of time. And so our ability to detect this is really now as there's a potential window. If we can detect the cancer while it's still located in the fallopian tube, then that way we can actually have surgical removal of the fallopian tube, preserving the ovary, and then preventing this widespread disease taking place.
Wow, that's incredible. Thank you so much for explaining that in layperson's terms, as I say. You know, I mentioned earlier that you were going to speak to liquid biopsy. Can you please explain what you call liquid biopsy and how you hope to apply that to ovarian cancer's early detection?
I'd be happy to. So one of the things I want to point out is historically, I want to go back and really point out where we are and why we need new cancer biomarkers. And when we talk about liquid biopsy, a lot of times we talk about blood, but we can talk about saliva, urine, you know, various bodily fluids that can be used for detection of disease. Well, in ovarian cancer, and I think probably everybody on this call has heard the term CA125 before, that is a protein or a serum marker that we can detect in the blood. However, that was detected by Bob Bass back in 1981. It's been 40 years since we've improved on biomarkers for ovarian cancer. Now, we've added some other biomarkers over the years, like HE4 and some shown over on the right side here, but they are not useful for screening. They're useful for potential for diagnostics. They're useful to determine benign versus malignant disease, but they're not very useful for overall screening because you can find CA125 elevated in certain patients that are individuals who don't have disease. They might have benign disease, might have something else going on. It doesn't detect everybody that has ovarian cancer. So that's why we really have worked on the idea of developing new approaches. And our approach is based off of, again, these examples of liquid biopsy where serum proteomics has been, or serum proteins have been the standard for many, many years. Steve and I have also worked in the area called circulating tumor cells, and those are cells that are shed by tumors that you can detect in the blood. There's work that's being done looking at DNA that it becomes basically released from tumors and that you can identify or enrich for that and sequence it. And we do that in my clinical lab quite a bit. So I also run a clinical diagnostic lab at our university within our cancer center. And we also have people who look at what are called microRNAs that are also in the circulation. That would be another marker that we could identify in here. And there's actually a group in Tina's Wish that is also studying microRNAs. What we study are called extracellular vesicles, or you'll hear exosomes quite frequently. And that is really what our focus has been. And I'll share a little bit why we're doing it and focused on this aspect. But again, blood tends to be the fluid we use for these liquid biopsies, but other fluids can be used as well. Now I'm going to show a little bit of this movie here just to kind of give you an idea about what we're talking about with exosomes. Now, exosomes can be, basically, this is a nice movie. You can actually see the whole thing on YouTube, but they're released by all types of cells. So this is where part of our challenge comes in. They're released by normal cells as well as disease cells. So we have a lot of work that we have to do to identify the ones that are cancer specific. And this is just a kind of a graphic kind of a version where you might have a cell here and they'll release these small extracellular vesicles or exosomes. These exosomes can get into the circulation. They can communicate between cells, but they can get into circulation. And one of the things that we're focused on is the idea that they actually have markers that are associated with the cell that they arise from. And what's shown here is the little spikes that you'll see on the surface of these extracellular vesicles. These can help us determine where they're coming from. And this is a lot of what our biology or research that we're doing is identifying these markers that are unique for specific forms of ovarian cancer. Now, I always have I get to escape that and then I'll go back to this. Now, just to kind of put that in perspective again, these cells release these exosomes. They have these specific markers that represent the cell of origin. We can look at these at really high power microscopes. These are called electron microscopes. And just to give you an idea of the size, they're 1,000 times smaller than the diameter of a human hair, but they're very abundant. And what I mean by that is, if we look just here in the middle, we can count the number of these circulating particles, and we can see an increase in the circulation based on stage. And what we have shown before here is that there's as much as a 12-fold difference in the number of these circulating markers between healthy individuals and those with ovarian cancer. And what that simply means, is it's shown here, is that there's over 2 billion of these particles that we're studying in a half a teaspoon of blood from an ovarian cancer patient. We might see like 16 billion in a normal individual. Well, it would say, wow, there's so many of these that are derived from the cancer itself. No, it's not. It's actually that there are many of these in the circulation because the body is responding to having cancer. And they start making, normal cells start actually making some of these almost like an immune response when you get an infection. So what our job is to tell the difference between what are coming from the cancer and what are coming from the host cells. And it's a little bit like Where's Waldo? I'm sure everybody's seen this somewhere along the way. Is our version of that is, where's the ovarian cancer exosome? And this is really what we have to do, is think that there's all these different circulating extracellular vesicles, but we're only interested in identifying these specific ones. And this is really an overview of our entire project that brings in the work of my lab here on the left, which is studying these markers on the surface of the ovarian cancer derived exosomes. Steve's work, which he'll talk about shortly, is the technology he's developed in order for us to be able to specifically capture these on microfluidic devices. And then the work of Claudio, who couldn't join us today, is really to now look at other markers where we can potentially take a very few of these and put them into another assay that amplifies that signal so that we can detect single molecules now that are coming from a single ovarian cancer cell at very high specificity and sensitivity. That means that we know it's from ovarian cancer and we can detect it in the very, very early stages, hopefully while it's still located in the fallopian tube.
Thank you so much. That was extremely informative, and I love the visuals as well. It really helps to understand what you're studying. Dr. Soper, maybe you can go into, what during your project, what has been the biggest aha moments or the most exciting developments for you?
Absolutely, Lauren. I'm first of all, I'm excited to be here with everyone this morning. So the big aha moments. Well, Andy, you can go to the next slide. The first one is, as Andy alluded to, is working with a very accomplished team of people with diverse backgrounds. So for example, a lot of people in the community think that medical doctors are the only people making contributions to managing patients with ovarian cancer and other type of cancers. And you know what? While that's true, it's only partly true. There's a lot of other people that are behind the scenes making important contributions to new technologies for managing ovarian cancer. And you're seeing two people here. Andy is a biologist. Claudio is a biologist as well. We have a team of clinicians that are working with us, and I'm a biomedical engineer. So I come at this from the engineering side of things, and that is what's really attractive, to bring together these diverse population and expertise to really move forward on new technologies that's going to help people who have ovarian cancer. The next big thing I would say is, just to kind of speak generally, is it's great to get new science published in journals, but I always tell my students, it's even a bigger compliment if you take those technologies and you get them to the bedside. That means you get them out so people can use them to help manage their cancer, ovarian cancer, for example. So that's where the engineers come into play. They take all these wonderful discoveries, as Andy showed, and he will be showing you, and try to make technologies that can do the measurements that are required to tell someone, do you have ovarian cancer or not, or any other type of cancer. So what you're looking at in this slide, there's a lot of science in here. And the big take-home message here is that we're developing a technology that can be produced in a very high mode, so we can disseminate this to the general population for screening. And that's extremely important to remember. And Andy alluded to this. Detect, and this is the major mantra for Tina's Wish: early detection, early detection. And the only way that that can be done, especially if you're asymptomatic, is to have at-home testing, and people, at least high-risk patients, are actually testing continuously to see if they have any signs or hints of ovarian cancer. And that's what you're looking at here. We've actually developed a test that uses a plastic chip, and that's shown in the upper left-hand corner. And this can be produced. We can produce millions of these chips per day and disseminate them into the community for testing. And as Andy alluded to, it's using a liquid biopsy. So what that means is just like if you're a diabetic, you're screening your blood sugar levels. The same premise can be used for ovarian cancer. We're taking a small blood sample, putting it into this chip, and then looking for those very small particles that Andy alluded to that are ovarian cancer associated, to see if that person has any hints or signs of ovarian cancer that may require further or follow-up studies. So this is the chip that we have produced that actually has, what I'm going to say, agents on it, on the surfaces of these pillars. You're seeing the array of pillars that are in this chip. There's 1.5 million pillars in this chip, and that'll actually have the ability to look specifically for those exosomes that Andy alluded to that are ovarian cancer associated. So you can go to the next slide, Andy. And we've used this technology already. We've been working in concert with Andy Godwin and his lab to get samples from metastatic, high-grade serous ovarian cancer patients and normals, and then use our technology to actually, from a blood sample, to actually see if they have ovarian cancer or not. And you can see for right now, what we've done is we've looked at in the slide or the panel that Andy is pointing to, we can actually distinguish between normals, non-ovarian cancer patients, and those with metastatic ovarian cancer. And that's what that basically slide shows you. And then in the lower left-hand corner, Andy talked about this electron microscope. Look at the size of the equipment that's required to make those measurements. It's untenable to move into the bedside. And what we have been able to do is to develop very small portable equipment that can go into a doctor's office or directly into a home for at-home testing, and with such ease of operation to allow very wide-scale screening. So this is actually quite interesting to us. And what we're doing right now is to look for early markers that are going to be indicative of early stage ovarian cancer, not just metastatic ovarian cancer.
Thank you so much. Wow, that's, I think, extremely exciting and feels like it's imminent that we're going to get this test. What are the next immediate milestones, and what do you need to reach those milestones, Dr. Soper?
Yes, absolutely. And I alluded to that briefly, Lauren. And one of that is we want to make our test specific for early stage ovarian cancer. Mm-hmm. And what that means is that now it can go out into the community as a screening test. That means everything can be done either at your doctor, your general practitioner's office, or even potentially at home. And what you're looking at here is, if you look at the middle picture, this is a cartoon of an instrument, a device that we're building right now and allowing you to do at-home testing for potentially for ovarian cancer. We have a commercial partner in this And this is very important because we want to get this out into people's homes and patients' use immediately. And the challenge here again is to make sure that we're only detecting those with ovarian cancer and not other diseases specifically. And then the other thing is the milestones that we need to do is to get this through the Food and Drug Administration for approval before we can ever disseminate this into the community for wide-scale screening. And that's a difficult task. Requires a lot of samples, requires hardware that's ready to go into people's laboratories for testing, but we're well on our way to do that. And then again, just to make sure that the test is highly specific for early stage ovarian cancer disease. And I'm going to just make one more little point, Lauren. This is not just for ovarian cancer. This can easily be repurposed for other diseases by simply changing the agent that we use that's looking for these exosomes. And I can give you a good example of that, where this test was actually first funded by Tina's Wish. We are now repurposing that for COVID-19 at-home testing by simply changing the affinity agent, looking at different particles, in particular the SARS-CoV-2 virus that generates COVID-19 disease. We can actually repurpose this specifically for at-home testing for infectious diseases. The virus particles are about the same size as the exosomes that Andy alluded to. And then we're looking at other diseases, endometrial cancer, a plethora of other ones that can use the same platform and with enough specificity to target those specific diseases. So lots of applications for this technology. That is for sure.
Wow, thank you so much. It's a lot to absorb. Dr. Godwin, can you speak to what you hope to accomplish in the next 12 to 18 months? And when do you think that women will see this early screen in their doctor's office?
So kind of going back to the sources of these extracellular vesicles, what are biological or scientific challenges, they are everywhere. They're in the foods you eat. They're in all these different kind of fluids that we talked about. So what we're really working on is this approach is to try to understand, and I actually have two of my students, my graduate students who are working in this area that are on this meeting today, is to look at these again, this colorful decorated surface of these, again, schematic of an extracellular vesicle, is to identify what each of these proteins are that decorate the surface. So these are proteins are our markers that we're focused on. So what we've been doing, and I should say we, or two of my graduate students are mainly Camille Trinidad, who's on here today, is that she has been identifying and isolating these EVs from different sources, from fallopian tubes, from ovarian cancer cell lines. And then she goes and basically working with a variety of different other expertise, tries to identify these specific proteins, what they are. So those are ways that we can then develop specifically those reagents to put onto Steve's chip. And so the more that we can have that are very specific for the ovarian cancer, the more specific Steve's chip will be. So this is what we're doing right now. And she has told me by the end of this month, we are going to have a lot of data that's going to help us identify more proteins that we can use to sort and basically enrich to tell us specifically that these are coming from ovarian cancer. Mm-hmm. Now, this becomes very scientific here, and I apologize to those, but what Claudio's work really is, and this has probably been the thing that's been the most challenging of our projects, but his group has really continued to work for this, is the idea of this assay that once we have these on Steve's chip, we can actually release. He has a mechanism that not only captures and counts the EVs on that microfluidic chip, we can collect those EVs afterwards, or those exosomes afterwards. They can flow through the microfluidic chip, we can capture them, and we can put them into another very sensitive assay that Claudio's building. And the idea behind that is that if you have no ovarian cancer seeds, In this case, those seeds are going to be ovarian cancer derived exosomes. If there are no seeds, you don't really get any kind of detection in his methodology. So over time, you might get a little bit at the later stages, but there's no formation of what we're looking for. And that's basically what are called amyloids. They're basically, what that means is a bunch of the proteins will come together and make these large organizations of proteins stuck together. So they'll remain separated. But if we actually put in a seed from ovarian cancer EVs, they now start to form these aggregates. And that's what's now being measured. And we can now use a device to measure that these seeds are being present, and they're forming these big aggregates that we can now measure. And so Claudio is working on this, and I'm hoping we've seen a lot of progress. We have monthly teleconferences or video conferences to go over each person's data, each person's group. And we've seen a lot of improvement and advancements just in the past 6 months with Claudio's project, which is really the one that helps us increase our sensitivity by magnitudes. And that's really what we're hoping to see.
Okay, given that, when do you think the early screening, not to put you on the spot there, but will actually be in the doctor's office?
Well, that's what, and that's why I think what Steve just shared with us was something that's really important. And that's the fact that we're working with companies now and technologies that can take our discoveries and move those into devices. And as you saw from that, Steve is working with a group now to build a device. So our job is to give him the best reagents to put onto that device. And I think that we'll really see a tremendous advancement towards the end of this year, refining those markers that we can use. And then we'll go through a validation study, which is very important. You have to then do a larger scale validation study to make sure what you're detecting is what you promise it to be, and we can detect it early, even before symptoms are arising.
So given that, will it be will there be a priority for high-risk women then?
Well, and I'll share a little bit of that with you here, is that one of the things we set up to help support this is our early detection screening program. So at our institute, one of the things I initiated a while back was really women who are coming in for their annual mammogram are giving blood samples. And we're then following those women over a period of time, which will provide us the samples of women who have came in at that point where they're doing their mammography, but they're also at risk of other changes in their health history. And so we followed those over time. So far, we followed them for about 8 years, 8 or 9 years. We have more than, I think we're now at about 5,500 women that are participating in this, who give us repetitive blood samples that we can then look and see when we can detect cancer in those individuals. Now, we already have 135 of these 5,000 women have been diagnosed with some cancer, including ovarian cancer. So this will allow us to look at how specific our test is. And then with the follow-up of what you asked about the at-risk women, I would say I'm very happy to announce that last week we got a very large grant from the National Cancer Institute that will help support the studies that we're doing here. And part of that money that we're getting from that grant is to help us build biobanks of samples from at-risk women. And shown here on the right are some samples that we have through a collaboration in Australia, where these are women that gave blood samples, and then within 3 months to 3 years, they were then diagnosed with ovarian cancer. Now, what I'd like to point out is the first 19 of these are from BRCA1 or BRCA2 mutation carriers, where the risk is substantially increased. I actually have been studying BRCA1 and 2 for a long time. I was involved in some of the early studies to map where that gene was before we even knew that there was a BRCA1. And so I've been involved in testing for BRCA1 and 2 for many years. But with this, we now have a consortium known as SIMBA that is following 60,000 BRCA1 and 2 mutation carriers. And working with them, we're starting to identify people who have blood samples of women who are not affected at the time they give a blood sample, but then later on develop that disease. And what I'd like to point out, why we're focusing on that, is 80% of all these women that we're doing from screening, 80% have high-grade serous tumors. So this is the disease we're trying to find. If you compare that to ones that you get of women who have the diagnosis of stage 1, the vast majority are not serous cancers. Only 20%, less than 20% are serous. This is the disease we're trying to identify. This is the most deadly form of disease. This is the most rapidly spreading form of disease. So that's where our focus has really been. So we have, with fortunately getting this grant just awarded last week, we are going to be able to provide funding to different groups throughout the globe to help us collect additional samples that will allow us to take the device that we're building and ask how far back can we detect disease before the patient was actually diagnosed. And so over here shows examples of that with the, for example, here, this sample was collected 25 months prior to a diagnosis, this patient 3 months. So we'll be able to go backwards and see how sensitive our test is, because for a screening test, you must identify cancer before symptoms. And that's what we're trying to do with this particular work.
Thank you, Dr. Godwin. Dr. Soper, what has been the biggest challenge or obstacles you faced with this project?
So a little bit of that is predicated on sample acquisition, because some of that recruiting patients was stymied because of the COVID-19 pandemic. The other big thing, and this is kind of a global challenge in any ovarian cancer research endeavor, is it's not only specifically finding the markers unique to ovarian cancer, especially those that show up in early stage diseases, but it's actually getting acquisition of samples of patients that have early stage ovarian cancer disease. Before I came to the University of Kansas, I was at the University of North Carolina, Chapel Hill, working with clinicians with ovarian cancer there as well. And the biggest dilemma that we had was finding people with early stage disease, because most of them are not diagnosed, to actually pull them through some of the trials that we needed to prove the technology was working. So that's been a big challenge. And the team that we've generated, and Andy had some good information on that, has really been active in getting samples across the entire spectrum, early to stage 1, stage 2, stage 3, and so forth. So this is going to be extremely helpful in terms of bringing our technology through the FDA to get clearance, so we can get it out so patients can start using it immediately. And that's what this game is about. We don't want to wait 20 years for this technology to end up into the bedside. We want to get this quickly into people's hands to use. And that requires a lot of work with the FDA, and our commercial partners are kind of facilitating that endeavor.
That's fantastic. Thank you so much. I'm just happy you're thinking about getting approvals from the FDA. That's like significant progress when I think about it. That's fantastic. Dr. Godwin, one closing question before we go into the chat or questions that people have. How can people who are participating today help or become involved in your work? Do you foresee a clinical trial in the near-term horizon?
Yeah, and so, there are several things. And first of all, education is always important, and to educate people to participate in studies. Now, we haven't started a study specifically where I've known some to reach out and try to work with advocacy groups to try to identify people who could participate by submitting actual samples. So that's one area that we're very interested in, again, is to identify, again, screening samples. We're involved right now, for example, our university, with a company that's doing colorectal cancer screening, and they need to, they're running a clinical trial that requires a lot of money and going to many institutes to collect a variety of different kind of samples. I think ultimately that's the kind of direction we want to go. But right now, I think with the grant that we recently got, it's going to allow us to at least get those samples in our hands that are going to be able to address the questions that Steve mentioned, from the fact of, we know we have a device that can distinguish between ovarian cancer patients and healthy controls. The ultimate, and the holy grail really is detecting the cancer before it's widespread and symptomatic. And so that's going to take a really team effort, because those samples are really, really rare. And there was a large UK study that followed about 300,000 women for 12 to 15 years. They're very interested in our technology and willing to work with us as soon as we show that in our sample sets that this is working. So that would be one of the biggest kind of collaborations that might come out of something like this, where we can connect with the large UK ovarian group and be able to take our technology, our assay, our biomarkers, and then apply them to a much larger sample set that we couldn't even begin to fathom to collect, because those require hundreds of millions of dollars over decades. And so, that's why the more we can demonstrate how sensitive and specific our test is, the more people are willing to work with us. And I think that's what the real excitement comes from. It was very interesting when I was walking in this morning, I ran into the head of our GYN surgical oncology group, who's part of this grant. And she was extraordinary excited. She said, what samples do we need to start collecting more that we're not currently collecting? And so, she goes, we're going to be looking at women with all different kinds of benign masses that will serve again as negative controls. And she's extraordinarily interested in expanding this, and then reaching out to her partners across the United States to also participate in the study. So, there's just word of mouth participation in letting, us know people who might be interested in doing that, reaching out to other institutes is really the way we're going. And I will say Tina's Wish has been wonderful because University of Pennsylvania is on this grant. And so Ronnie Drapkin and a good long-term colleague. So for people to know, I was at the Fox Chase Cancer Center in Philadelphia for 26 years. So I was on the East Coast for quite a while before I came back home to Kansas. And I've been able to establish, go back to my old biobank at Fox Chase. I've gone to my colleagues at University of Pennsylvania, who are now part of this grant. And we're just basically, expanding our reach so we can get those kind of samples that, first of all, tell us that we can detect cancer 100% of the time and knowing it's ovarian cancer.
Thank you so much. Thank you. What a fabulous presentation today, and we really appreciate all the information you shared. I'm mindful of the time, so I'm just going to entertain one question from the chat function before we close out. Given what you just described, and somebody asked a question as a follow-up, will the women who are determined to be at high risk for developing ovarian cancer have access to your screening techniques sooner than the general population, do you think?
Yes, and I really think that, in general, if you do just general screening, because of the rarity, ovarian cancer is still considered a rare disease. But, as I mentioned before, I have been studying BRCA1 before it was even cloned. And I have followed many high-risk families for many years, and that really is the population we want to do. With mammography and breast screening, we can identify women who have a BRCA1 or 2 mutation or high risk of breast cancer. But it's very invasive for ovarian cancer in those same women, and they tend to basically undergo oophorectomies and have their ovaries removed completely as a preventative factor. So our goal really is to be able to identify those women who are at high risk and potentially offer them, again, ways to prevent that development of ovarian cancer. And again, they're at significantly high risk compared to the general population when you're a mutation carrier. And so that would be the population that we're really focusing on. And that's where I'm trying to really leverage I don't know how many years, it's been at least 15 plus years I've been part of SIMBA, really leverage a lot of my colleagues that represent 52 countries that are studying more than 60,000 of these carriers, is really leverage my kind of connections there to keep developing those kind of tests in the high risk population through that organization. So I think that but it's something we have to bring to them and, show them that this is going to work and that, they need to, help advance what we're going to do, but clearly it's going to be initially in the high risk populations. And then, as Steve mentioned, if you can make this test cheap enough, and at a physician's office when a woman comes in for their pap smear, get one of the, we don't even have a name for it yet. I guess we got to figure out a catchy name, but get a test done at the same time they're getting a pap smear. Something like that would be really the ideal situation.
Again, thank you so much. I love that we're even thinking about maybe naming it at this stage. So, thank you so much, and for your presentation, and thank you everyone in the audience for participating and attending this morning's virtual researcher roundtable. A huge thanks to Dr. Godwin and Soper and Soto for their groundbreaking work. Beverly will follow up with a recording link of today's conversation, and to solicit your input and participation in Tina's Wish, please share this link with others. We love to share our knowledge and efforts, and would like more participation. So please feel free to send it to whoever you think might be interested. Tina's Wish plans to feature several researcher roundtables this year, so stay tuned for the details regarding our next one. Each one will be a little different. And lastly, don't forget to register for the Empower Me series. Links are in the chat box, and have a fabulous day, everyone. Thank you.
Thank you.
Thank you.